Genetic characterization of a PRRS-1 strain emerged from recombination between two MLV vaccine strains. My name is Kvisgaard. The talk will be divided into two parts. Part 1 will be the description of the PRRS-1 outbreak in a PRRS-negative bore station in 2019, and the second part will show the results obtained from an experimental study of the efficacy of free MLVs upon challenge with the recombinant PRRS-1 strain isolated from the bore station. In July 2019, routine surveillance of blood samples from a PRRS-negative bore station was found PRRS antibody positive. Screening of new samples by real-time PCR showed that the farm was indeed infected with PRRS-1. We continued to do the O5 sequencing on selected samples to determine which strain had infected the bore station. Here is shown a phylogenetic tree with Danish and globally representative PRRS-1 O5 sequences. We see in the bottom of the tree the so-called Danish cluster that also include the very first detection of PRRS-1 in Denmark. In the top of the tree we have the porcelain-like sequences, and here in the middle of the tree we see the results from the sequencing of the samples obtained from the bore station, now called the Horsens virus. We zoom in on the Horsens virus, we see that the samples are very similar to the unistrain, which is the strain used in the Emavac PRRS MLV vaccine. We continued with sequencing of OF2 through OF7, which is the part of the genome that encodes the structural proteins. We did a pairwise nucleotide comparison of the Horsens virus sequence to the unistrain vaccine strain sequence. And we see here that in O4 through O7, the Horsens virus sequence is very similar to the unistrain. Whereas in O2 and O3, we see a decrease in the similarity of the Horsens virus sequence to the unistrain. The closest match against OF2 in GenBank was to a Belgian strain from 1994, and they shared approximately 93% identity. We continued with a full genome sequencing of the Horsens virus by NGS, and we confirmed that the 3' part of the genome of the Horsens virus was very similar to the unistrain, whereas the majority of the sequence of the Horsens virus was of unknown origin. The closest match to this unknown part in GenBank was to the Lillestad virus, but only with 92% similarity. So is it a recombinant strain that had infected the bore station? We received samples from a neighboring farm and screened the serum samples for PRRS by real-time PCR, and we determined that the farm was PRRS-1 positive. Selected samples were sequenced in OF2 and OF5. Here is shown the results from the sequencing of the samples from the neighboring farm, and in some samples we found sequences that were unistrain-like, that is, that they are very similar to the unistrain both in OF5 and in OF2. But other samples from the neighboring farm were found to be 96V198-like, shown here in the bottom of the screen, both in OF5 and OF2. And then we also found that the Horsens strain, which is shown that in OF5, the strain from the neighboring farm is very similar to the Horsens strain and also to the unistrain, whereas in OF2, it's very similar again to the Horsens strain, but also to the 96V198 strain. This 96V198 strain is the strain used in the vaccine MLV vaccine. So could this strain be the major parent of the Horsens virus. To determine that, we did a recombination analysis, and here is shown the results. We have the Horsens strain shown in the red line, and in the 5' end of the genome, we see that the Horsens strain follows the blue line, which represents the 96V198 strain. In the 3' end of the genome, the Horsens strain jump and start following the green line, which is represented by the unistrain vaccine strain. So from this analysis, we confirmed that the Horsens virus indeed is a recombination between the two vaccine strains, where the major parent is from the vaccine and the minor parent is from the unistrain vaccine. We also determined that the breakpoint was situated in OFF3. We were later told that the neighboring farm had actually used both the vaccine and the unistrain vaccines prior to the outbreak in the boar station. We don't know how far apart the vaccines were used or whether they were used in the same animals. But we did, from our results, find that the vaccine and the unistrain vaccine strains were circulating in the farm, and also detection of the recombinant Horsens strain. If we look at the location of the boar station, it's shown here on the map in the right-hand side, and the neighboring farm is shown on the left-hand side of the map. And there is approximately 6 kilometers distance between the two farms. So the question is PRRS airborne over 6 kilometers? We don't know, but if we look at the data obtained in the weeks prior to the outbreak in the boar station, we see that the wind direction was actually from the west and southwest. So it may be a possibility that PRRS was infecting the boar station through air. The impact of the Horsens virus in the field was that more than 40 farms were infected through semen from the boar station, and we were also told from field veterinarians that they saw much more severe clinical signs in sows and an increase in piglet mortality than normally seen upon a PRRS-1 infection. The impact of production losses in affected sow herds has been published by Christensen and co-workers in 2020. The question now is this Horsens strain more violent than other PRRS-1 strains, and will existing vaccines protect against it? And so that question leads to the second part of the talk, where the aim of the study was to investigate the efficacy of 3 different commercial PRRS-1 MLV vaccines upon challenge with the recombinant strain, and to compare the pathogenicity of the recombinant Horsens strain with the typical PRRS-1 field strain. In Denmark, 3 PRRS-1 MLVs are on the market, where the most often used vaccine is the Porcellus vaccine, followed by the unistrain vaccine, and only rarely is the vaccine used. On the right-hand side, you see a phylogenetic tree of full-length PRRS-1 sequences, and the 3 vaccine used in the study is marked here with red arrows. We have the Horsens strain here with blue, and the reference strain used in the study with green. In the study, we used 36 4-week-old pigs that was divided into 6 groups, with 6 pigs in each. We had 3 vaccinated groups, one group that was vaccinated with the vaccine, one group that was vaccinated with the unistrain vaccine, and one group that was vaccinated with the Porcellus vaccine, and then subsequently challenged with the Horsens strain. Then we had two non-vaccinated groups. One group were challenged with the Horsen strain, and one group were challenged with the reference strain. And then we have a negative control group where the pigs were left without any treatments. Pigs were vaccinated at DPV 0 and challenged 28 days after vaccination. Blood samples were collected 3 days before vaccination and then subsequently once a week after vaccination throughout the study. Pigs were euthanized at day 28 post-challenge, that is 56 days post-vaccination, and tonsils, lymph nodes, and lung tissues were collected from the pigs. No respiratory clinical signs were observed throughout the study. During the first week post-challenge, a few pigs in the non-vaccinated but challenged with the Horsen strain group had a fever. This group had also a significant decrease in average daily body weight gain in the first week post-challenge when compared to the vaccinated group. At necropsy, slightly enlarged and congestive lymph nodes were observed for most of the infected pigs, but no lung lesions were observed in any of the pigs. Looking at the viremia, we see here that at day 7 post-vaccination, all the vaccinated pigs were positive in PCR. We see that the POSI group peaked at day 7, whereas the Suvaxine and the Unistrain groups peaked at day 14 after vaccination. Upon challenge, an increase in viral load was seen in all 3 vaccinated groups at day 8 after challenge, and then it was followed by a decrease in viral load until the end of study. For the non-vaccinated but challenged with the Horsen's virus, we saw a peak 8 days after challenge in the viral load, and for the reference group, we saw the peak 3 days after challenge. For the viral load for the Horsen's group at 3 days post-challenge, we saw a significant higher load compared to all the 3 vaccinated groups. And at day 8 after challenge, we saw a significantly higher amount of virus in serum in this group compared to the 4 other groups in the study. Do we look isolated on the viral load after challenge? We see that the Horsen's group had a significantly higher viral load compared to all the vaccinated groups and also to the reference group. Among the vaccinated groups, the POSI group had a significantly higher viral load compared to the Suvaxine and the Unistrain vaccinated groups. Looking at the viral load in tissue, we see that the highest viral load for all groups were detected in tonsils. The viral load in lungs were varied between groups, but no significant difference were detected. Only few clinical signs were observed after challenge. We saw a significantly higher viral load in serum for the non-vaccinated but challenged with the Horsen's virus group compared to all the other groups. Only limited differences in viral load in tissues between groups and there was no indication that the recombinant Horsen strain should cause severe disease when compared to other known high pathogenic strains. But in this study, we saw that all 3 vaccines used had a significant impact on the viral load in serum after challenge with the recombinant strain. An assessment of the impact in challenge in sows has also been conducted by Genesis et al. And published in 2020. So the current status is that the recombinant virus is still out there in the Danish pigs, and it is drifting. We will keep sequencing diagnostic submissions to monitor the viral evolution of this new recombinant strain. I would like to thank my co-authors and colleagues from University of Copenhagen, ANSYS, SEGES, and Technical University of Denmark. I'd like to thank you for your attention. For any question, please contact me by email. Thank you very much.